Okay, so this is the electron transport chain and fermentation presentation. I'm Abigail Lister.
I'm Karen Parr. I'm Haley Alex.
I'm Savannah Drury.
I'm Amaya Tovar.
Okay,
All right.
so microbial metabolism. Microbial metabolism refers to the set of chemical reactions and processes that microorganisms use to convert nutrients into energy and building blocks necessary for growth, reproduction, and maintenance. These processes are crucial for survival and vary depending on the available energy sources, environmental conditions, and the type of microorganism. For catabolism, the breakdown of complex molecules like sugars, lipids, or proteins into simpler molecules like glucose, fatty acids, or amino acids to release energy, typically in the form of ATP. For example, glycolysis and the citric acid cycle are catabolic pathways that help microorganisms generate energy. Anabolism is the use of energy to build complex molecules like proteins, nucleic acids, and lipids from simpler precursors. This is an energy-consuming process that's essential for cell growth and reproduction. Microorganisms can utilize different energy sources and metabolic pathways depending on the environment, such as aerobic respiration using oxygen, aerobic respiration or anaerobic respiration without oxygen, or fermentation, which can occur in the absence of oxygen, produce energy. The type of metabolism a microbe uses affects its growth rate, environmental adaptability, and interactions with other microorganisms. Um, for a glucose containing media and why it's important, you need to understand what a culture media is in general, which is a nutrient-rich substance that's used in labs to aid in the growth and multiplication of microorganisms. Glucose is a simple six-carbon sugar. It's the most abundant monosaccharide and is similar to heavier amino acids of the same size. It can cross plasma membranes with facilitated diffusion and transport proteins. This makes glucose the primary fuel source in cell culture. Controlling the medium in a scientific setting, whether it contains glucose or not, helps scientists study the different metabolic processes of microorganisms. If there's too little glucose, the microorganisms will starve. Lack of glucose can also lead to a hostile environment where endospores can form. Endospores are resistant to many things and can change the results of different studies. If the glucose is too high, that can also have a detrimental effect on many different cell types. Glucose-containing media matter because they provide an essential energy source for growing and studying cells or microorganisms in lab settings, and manipulating the glucose levels can reveal insights into cellular processes.
Electron transport change of E. coli, glycolysis and Krebs cycle. The electron transport change of E. coli coupled with glycolysis and the Krebs cycle plays an important role in cellular respiration and ATP production. Electron transport change is found in E. coli inner membrane and it transfers electrons to oxygen or other terminal electron acceptors from NADH and FADH2, which are created during glycolysis and the Krebs cycle. The electron transfer produces a proton gradient, which stimulates ATP synthesis during oxidative phosphorylation, and this this action generates a proton motive force, which is required for ATP generation because protons return to the cell through ATP synthesis. Glycolysis in the cytoplasm, glycolysis breaks glucose into two pyruvate molecules, producing two ATP and two NADH. Energy extraction is started by the mechanism, which also provides Krebs cycle substrates and allows ana- anaerobic fermentation. Krebs cycle happens inside the cytoplasm and turns pyruvate from glycolysis to carbon dioxide while creating NADH and FADH2 for the electron transport change. Each cycle turn produce 3 NADH and FADH2 and 1 ATP, which are important to the cell energy's requirements.
For the electron transport chain, the electron transport chain starts when electrons are passed through the series of protein complexes in the mitochondrial membrane. It's located in the plasma membrane of E. coli because they lack a mitochondria. For the electron source, electrons are provided by reduced cofactors like NADH and FADH2 produced from glycolysis and the citric acid cycle. For the protein pumping, as electrons move through the protein complexes in the electron transport chain, energy is released. This energy is then used by specific protein complexes to pump protons from the cytoplasm to the periplasmic space, which is the area between the plasma membrane and the outer membrane. The main protein complexes and the mobile electron carriers are NADH dehydrogenase, cytochrome B1 complex, cytochrome C, and cytochrome C oxidase. The proton gradient is the active pumping of protons into the plasmic space, generates a proton gradient across the plasma membrane. The use of the proton gradient is to store energy, and the energy is used by ATP synthase to synthesize ATP as protons flow back into the cell through ATP synthase complex. This process is known as chemiosmosis. Protein pumping is important because it creates necessary electrochemical gradient for ATP production.
Fermentation. E. coli is a facultative anaerobe, meaning it can adapt to aerobic, anaerobic, and fermenting growth conditions. And for microbes like Streptococcus pneumoniae, fermentation is its only option due to the lack of an electron transport chain. The purpose of fermentation when glucose is involved is to take the NADH produced from glycolysis and to turn it back into NAD plus so that the glycolysis cycle can continue. There are also many different fermentation pathways, all starting with the pyruvate from glycolysis. So they all have different outcomes. The mixed acids pathway involves microorganisms like E. coli to reduce NADH to NAD plus, and in doing so, various mixed acids are the byproduct, like lactic acid, succinic acid, ethanol, acetic acids, and gases. These pathways are important because it allows cells to continue making ATP in anaerobic conditions.
And then just a Christian perspective in healthcare, I chose the quote, each of you should use whatever gift you have received to serve others as faithful stewards of God's grace in its various forms. So what I put is healthcare personnel are called to be good stewards of human health by using their skills to serve others, patients, with compassion and excellence. The Bible teaches us that caring for the sick and suffering reflects Christ's love and fulfills his command to care for the least among us. Healthcare workers play a vital role in bringing healing, comfort, and hope, therefore acting as God's hand in the world. The service of healthcare workers is not just a professional, but a divine calling to uphold the sanctity of life and demonstrate God's grace through compassionate care.
Awesome. And then these are our references. So that was the electron transport chain with E. coli and fermentation. Hope that helped.